Hello students, I'm Dr. Arpita De. Welcome back to the second part of our apoptosis lesson. In the previous lesson, I had talked about what is apoptosis, what are the causes and consequences of apoptosis. Now here in the second part of the lesson, we will be talking about the underlying mechanism of apoptosis. Now here, what we can see in this picture is an old man who is absolutely confused about the term caspase. He's thinking, what are caspases? Now let's help him to find out what are caspases. Okay? Caspases are cysteine aspartate proteases. That means what? They are cysteine dependent aspartate directed proteases. Okay, there are two types of caspases, the initiator caspase and the executioner caspase. Fine. Caspase 2, 8, 9 and 10 are the initiator caspases while caspase 3, 6 and 7 are executioner caspases. All caspases remain in an inactive form known as procaspases. These procaspases are cleaved and converted into an active form by intrinsic or extrinsic pathway. Okay, the executioner caspases are activated by initiator caspases. These caspases contain a prodomain. All right, this prodomain contains caspase recruitment domain, which is known as CARD. Now we'll talk about the pathways of apoptosis. Fine. First, we'll talk about the extrinsic pathway of apoptosis. When we tell the word extrinsic, a feeling comes to our mind that there is something to do with external agencies. Okay, when there is an external signal or a signal that is coming from outside the cell, we call it or we classify it under extrinsic pathway then. Fine. Here, what happens in extrinsic pathway? Let's talk about that. Extrinsic pathway has got two subdivisions. One is the Fas pathway and the other is the TNF pathway. We'll talk about Fas pathway in detail over here. Okay. In Fas pathway, what we see, the external stimuli is in most cases a cytokine. Okay. The most studied cytokine that induce extrinsic pathway is an extracellular messenger protein called TNF. Alright, TNF cytokine is produced by the cells of the immune system in response towards adverse condition, that is, adverse condition provoke immune cells to produce TNF. Fine. Now, Fas receptors are present on the surface of the target cell. Alright. Now, the Fas ligands are there on the killer lymphocyte. Fine. I had jumped one step of diagram over here. When the ligands are not yet bound to these receptors, they are free in nature. After the ligand binds to the receptor, there is a trimerization of receptors. Okay. After trimerization, what happens? There is a conformational change in the receptor, and this recruits FADD. We call them FAD. Now, what is FAD? FAD is fast associated protein with death domain. It's also called MORT1, which is an adapter protein that bridges members of TNF receptor superfamily to form death-inducing signaling complex or DISC during apoptosis. Okay, FAD has got DED, which is known as death effector domain. These death-affected domain, along with fat, they form disc, which is known as death-inducing signaling cascade. It is formed by accumulation of death domains that get activated by Fas ligand binding. Thereafter, by virtue of these dead domain or DED, it recruits the inactive procaspase 8. Look at this. Look at this part. FAD has got this DED, these three black balls. Okay. Now this one, this striped structure, they are the inactive procaspases or inactive procaspase 8 more precisely. Alright. These inactive procaspase 8s are recruited by virtue of their DED to the FADD. Now what happens? They are getting converted to active caspase 8 by cleavage. See, it's written over here. Inactive procaspase 8 is cleaved to active caspase 8 that further cleaves procaspase 3 to caspase 3. This procaspase 3 is also inactive. And this caspase 8, once activated, goes and cleaves this procaspase 3 and converts it into active caspase 3. Alright, this active caspase 3 goes on to inactivate the inhibitor protein that remains associated with nuclease enzyme. Alright, the nuclease is now free to destroy the nucleus and DNA. As long as this nuclease is bound by an inhibitor, what will happen? They will not be able to reach out to the genetic material and degrade them. But once it is made free, it can easily travel to the nucleus and destroy the DNA. This is the brief summary of the extrinsic pathway of apoptosis. Fine. Now we look at the cascade in detail. Procaspase 8 is initially inactive here. This is made active by the phenomenon of cleavage. Fine, it is cleaved and converted into an active caspase 8. And this active caspase 8, what does it do? It goes and cleaves procaspase 3 to convert it into active caspase 3. This then goes and what does it do? We had just now discussed. It inhibits the nuclease enzyme inhibitor. The inhibitor gets destroyed. Nuclease enzyme is free to attack the nucleus, and finally what does it do? Nuclease enzyme destroy the DNA in the nucleus. This is the cascade of destruction. Fine. Now we'll talk about the intrinsic pathway. What is causing this intrinsic pathway to take place? This intrinsic pathway takes place when there is severe DNA damage, or we can also call it genetic damage, when there is lack of oxygen or very high concentration of cytosolic calcium, or there is a presence of viral proteins or severe oxidative stress. I think you all know what is oxidative stress, formation of reactive oxygen species and all. That is oxidative stress. Okay. Now, this intrinsic pathway is facilitated by certain families of proteins. What are they? These are pro-apoptotic proteins, anti-apoptotic proteins, and BH3 only proteins. First, let's briefly see what is pro-apoptotic protein. Pro-apoptotic proteins are those which promote apoptosis in normal cells. Examples, bats, etc. Okay. Then we'll come to anti-apoptotic protein. These anti-apoptotic, from the word itself you can understand, anti-apoptotic proteins are those which inhibit apoptosis and ensure cell survival. Examples are BCL2, BCLXL, BCLW, etc. And then comes the BH3 only protein. These proteins only have a small BH3 domain and they inhibit or promote apoptosis through indirect mechanism. Okay, they either inhibit anti-apoptotic members or stimulate pro-apoptotic BCL2 members. Examples are BID, BAD, BIM, etc. All these three, they they fall under the broad subhead of BCL2 family protein. Now let's see what is intrinsic pathway. Intrinsic pathway is also known as mitochondria mediated death pathway. It happens as an answer to or as a reaction to signals that come from within the cell. Okay, here what happens, let's see. Here in intrinsic pathway, this intrinsic pathway is activated by intracellular signals generated when cells are stressed and depends on the release of proteins from intermembrane space of mitochondria. Okay, mitochondrial swelling takes place and there is leakage of cytochrome C. There is leakage of cytochrome C from the mitochondrial intermembrane space to the cytoplasm through pores that are generated on the membrane. All right. What happens now? As this cytochrome C uh, reach the cytoplasm, it interacts with APAF1. Now, what is this APAF1? These are special proteins. These are adapters, you can say. All right, these are specialized adapters. Apoptosis protease activating factor. Okay, these are disc-shaped heptamer, or we will see that they help in the formation of wheel of death. All right. Mitochondrial proteins or SMAC, which is known as SMAC. Okay, second mitochondria derived activator of caspases are released into the cell cytosol following the increase in permeability of mitochondrial membrane. SMAC binds to proteins that inhibit apoptosis, that are BCL2, BCLXL, and BCLW. Ultimately, what happens then? These cytochrome C reaches the cytoplasm, it interacts with APAF1. APAF1 then oligomerizes and interaction with procaspase 9 forms an apoptosome, not apoptosome, okay? Apoptosome, which is a cartwheel-like structure. This apoptosome is also known as the wheel of death. Once these procaspase 9 gets associated with this APAF1, it simultaneously undergoes the procedure of cleavage. It's cleaved from an inactive procaspase 9 to an active caspase 9, which is now going to execute apoptosis. All right. Now let's see what is the relation between these two. Here, see again, it's like a brief recapitulation. Here, the fast receptors are there, which on binding of ligand trimerizes, there is a conformational change. Okay, it recruits FADD, and by virtue of this DED domain, it recruits inactive procaspase 8. Then they form the disc or death-induced signaling cascade. All right, then this inactive procaspase 8 is converted to active caspase 8. This active caspase 8, what does it do? it converts bit into truncated bit. Now this T bit or truncated bit goes and activates backs and back, which are responsible for making pore on mitochondrial membrane. Once the pores are formed on mitochondrial membrane, there is release of cytochrome C. The cytochrome C then associates with that APAF1 and forms the apoptosome. This formation of apoptosome leads to the activation of procaspase 9 to active caspase 9, which ultimately allows apoptosis to occur. And from here what we see, this active caspase 8 goes on to convert inactive procaspase 3 into active caspase 3. This activated caspase 3 conducts the execution, that is apoptosis. So we can see that these are not completely independent processes, but they are interlinked. This T bit is playing the role of a bridge that connects these two processes. All right. Now, in a nutshell, what we can say, what is the take-home message? It's a tightly controlled cell death program. It takes place when cells lack survival signals or where there is extensive DNA damage. Then, conditions leading to apoptosis are both physiological as well as pathological. Then it involves two mechanisms, extrinsic and intrinsic pathway. And finally, the study has immense application in medical science, especially cancer biology and also developmental biology. Hope you had learned something new. If you have any doubt, any questions, do not hesitate to write me in the comment section. Thank you so much for your kind attention. If you like this video, don't forget to hit the bell icon to subscribe my channel and get updated as and when there is a new post in the channel. Thank you and goodbye.